So low density lipoprotein, what does that mean?
It doesn't have high density lipoprotein.
So what is it that makes it less dense?
Low density?
Mm-hmm.
It has more lipids and less protein. I don't know if that's all what you're looking for.
Yeah, no, that's exactly what I'm looking for. And I'm trying to find the picture. There it is. So VLDLs is how they start, usually. And then we end up with our, so VLDLs, and then we end up with our LDLs. So the job of an LDL is literally to float around the bloodstream and dump off lipids to tissues that need it for energy, or that need it to make more cell membranes, right? Because these are just, they're lipids, they're phospholipids. We use, there's phospholipids and then other lipids inside. But the idea is it's transporting, it's dumping off those lipids to be used by the cells. When there's extra, we got to put it somewhere. It doesn't just keep floating around, it's going to dump it off somewhere. And that's what it ends up in your arteries, in your gallbladder, in places you don't want it.
So HDL is what we want?
HDL's job is to go around and pick up the extra.
Okay.
So you want a good, you don't want, you want both, but you want, really they're looking for a ratio here. You want a good ratio of HDLs to LDLs so the HDLs can pick up the extra so it doesn't end up in your arteries and whatnot. Extra lipids.
Extra lipids.
Yeah.
And low LDL. What was that one again?
So LDLs drop off lipids, HDLs basically pick them back up.
So it's good that the LDLs, it's good that they're dropping off the lipids to the cells, but it's just because they have a lot of excess and they get
Right.
to places where they don't belong.
Right. Yeah. It's when we take in so much lipid, more than we need, we have to do something with it.
Okay.
So we call them low density and high density. Anybody know why that is?
Because of the protein lipid ratio.
Yeah, exactly. So proteins are more dense than lipids, right? Lipids float on water, proteins would typically sink. So low density lipoproteins have more lipids. High density have more proteins. Good, you got that. So all but the lipids. So that table that I showed you, with all the different enzymes, that's part of that question.
Okay.
But so everything but lipids, so it gets broken down like that table shows. And then once it's small enough, the whole idea is we have to get it small enough that those microvilli can absorb it.
Mm-hmm.
So once they get small enough, then they just absorb it straight into the blood. The proteins and the carbs go straight into the blood. Problem with lipids is they're lipids, they're not water soluble, and blood is mainly water. So when we did the lymphatic system, it said the function of that was lipid absorption. So in the villi, let me get the blown up picture so you can see it better. Maybe.
So they all have the same answer except for lipids, right?
Basically, the enzymes are going to be different. What causes them to release are going to be different, but the absorption part is the same. So this is figure 2416. So if this is a villus, right, just one of these villi, literally that gets absorbed straight through the villi right into the blood. Lipids can't do that, like I just said, so they have to go into the lymphatic system, into these lacteals. So we package them into these. Let's just go to the diagram.
So that was like
Exactly. So let me get to that diagram real quick. It's in here somewhere. So like I said, glucose or proteins come in, we transport them right into the blood. Lipids, on the other hand, so figure 2430 for me, we make micelles first. That helps get them through that initial cell layer. Micelles, M-I-C-E-L-L-Es. So that's what we talked about. So bile gets secreted, right, from the gallbladder. So bile emulsifies lipids. What that really means is it takes those big droplets and breaks them into smaller droplets. So first we emulsify it, making it smaller, and then bile salts. Salts are water soluble. So these salts surround that lipid and make it water soluble enough to pass through this membrane. So if this is the cell of our villi right here, right? Really, it'd be, so this would be one of the cells in a villi. So we put bile salts on it. That gets it into the cell. Once it gets into the cell, it's just a triglyceride, just lipid now. So then we take a bunch of proteins and stick it on it so that it'll pass out of the cell and into the lacteals.
So you said that the bile emulsifies it, so it breaks it down, and then bile salts surround it,
Mm-hmm.
make it soluble to pass through the membrane.
Exactly.
And then once it gets in there, protein attaches to it?
Yep, because the bile salts stay behind basically. They don't cross into the cell, but they help the lipid get through the cell. So then we put those proteins on it, we put it into the lacteals, and it goes through the lymphatic system, and part of that process is now that lipid is going to become part of a VLDL, LDL, and that's how we actually get it where we need to in the body.
Okay, so the protein attaches to the lipid, goes to the lacteal, which is in the cell, or?
It's in the villi.
Oh. And then from there it goes into the cell?
Yep, so the lacteal is basically just a lymphatic capillary that's inside of a villus, or inside the villi.